We are having this special publication Q&A session with Dr. Rajendra Kumari, Executive Director of Integrated Services at Crown Bioscience. And we are reviewing the recent publication entitled A Living Biobank of Matched Pairs of Patient-Derived Xenografts and Organoids for Cancer Pharmacology. And now our first question for Dr. Kumari related to the publication. Can you tell us about the Living Biobank and what is the importance of patient-derived models for cancer?
So patient-derived models are more predictive of the clinical response than standard cell line models. And these, so both PDX and organoids, which are derived directly from the patient tissue, are different from the human cell lines because they haven't adapted to grow in plastic, in 2D. They haven't been maintained in this artificial environment for many sort of passages. And the 2D cell lines don't actually resemble the patient. The patient-derived models preserve the original patient tumor histopathology and the molecular pathology, and therefore these models are more reliable for predicting clinical response. We have a vast collection of PDX models which represent patient diversity and heterogeneity, and these are widely used for mouse clinical trials, for translational studies, to inform on patient stratification, for potentially identifying biomarkers. And so what we've done is we've harnessed the cancer stem cells from these PDXs, which are, we have in our living biobank, and use specific organoid methodology to create a large collection of new organoids. So we have over 500 different organoids, over 15 different cancer types. And we refer to these as PDXOs because they're derived from the PDX. And the living biobank preserves the original tumor features, and also the models can be used over and over again because they've been cryopreserved and they can be reanimated back into culture and into mice. So this diagram in figure 1 is a schematic sort of workflow of the process for creating the organoids, for biobanking them. And then we characterize them for histopathology. We perform next generation sequencing, compound testing, as well as QCing for authentication and comparison back to the original tissue. And in this figure, we show how the histology, for example, in pancreatic cancer, lung cancer, and melanoma is maintained in those 3D organoid cultures. And also on the right-hand side, we demonstrate how the whole exome sequencing and RNA sequencing across many different models, which are represented by the dots, have high correlation to the original tumor of origin.
So what is the advantage of having organoids derived from the PDX?
So this strong correlation genomically and with histopathology between the PDX and the organoids means that they are biologically equivalent, just like the organoids are with the original patients, and the patient-derived xenografts are biologically equivalent to the patients. And by harnessing the cancer stem cells with the organoid protocols means that you retain all those original features of the source material. Now, you can say the same for cell line systems or other 3D systems which may use material like PDX. But the difference is in the predictability of response, which is shown in the pharmacology studies we did in figure 3. So in the table at the top here, we're summarizing the IC50 response and the in vivo tumor growth inhibition for EGF inhibitors. And this is in one of our non-small cell lung cancer models, LU1235. So this model has been extensively used as PDX because of the EGFR activation mutation, so it's got exon 19 deletion. And we have a lot of in vivo pharmacology profiling with this model, which has been published as well. And it's very responsive to EGFR inhibitors. So the TGI shows that there's tumor regression, which is summarized in the right-hand side of the table. And then in figure 3A, which is a typical in vitro IC50 generated with a dose response curve in our organoid model, again, the response to the EGF inhibitors shown here and also summarized in the table, you can see that the IC50 value is in nanomolar range, which translates to that tumor growth inhibition showing a tumor regression. So this is a highly responsive model, both in vitro and in vivo. So what we wanted to do was look at many different compounds, such as chemotherapeutics, targeted agents, both large and small molecules, determine the correlation of the in vitro response to the in vivo response. And this was done over a number of different cancer types, and this is shown in figure 3b. And the overall prediction we found was 86%, and this was higher than other systems such as the 3D ex vivo models, which uses PDX tissue. So this means that more in vitro drug discovery using organoids can be performed with confidence, knowing that the responses observed can be translated in vivo, and then the same models can be used from early sort of discovery work to late translational studies. And also with the recent FDA Modernization Act using alternative technologies to animal models, organoids will have a huge impact here because of their predictive potential and allowing or making early drug discovery preclinical decisions more relevant to the patient.
And how are these organoid assays validated for in vitro screening?
For in vitro screens, the organoids are expanded first, working from our sort of working biobanks, and it plated into 384 well format. The expansion phase can be dependent on the model growth, which is characterized during that biobanking stage, and only models that show consistency in growth are ready for service. The 384 well assay format has also been validated so that the assays are reproducible and robust for high throughput screening, using multiple drugs and multiple models. So that gives you, confidence in the results. So just like any sort of standard 2D cell screen for viability to measure tumor killing, as an example, using CTG, we have evaluated the signal to noise ratio in the assays, which is shown here in figure 2A. We've looked at the Z prime factor, so there's a medium of 0.63, which is indicative of a good assay and above industry standards, and also we show consistent IC50 values across experiments, which again assesses the interplate variation. And this is very comparable to some of the 2D assay systems as well. Now, we did find that the organoid assays were more robust and reproducible than, let's say, using sort of standard fresh tissue from PDX in ex vivo type assays, and also more efficient in terms of expansion and assay preparation. So this meant that we could screen those multiple compounds with multiple models swiftly. And as an example shown here, with the waterfall plots, you can see that the response across collections of models could be generated representing patient diversity, and we could then compare them side by side, and this wouldn't have been possible with PDX models or even ex vivo assays.
And by chance, can you tell us the key differentiator of Crown Bioscience biobanks?
Breadth and depth of characterization of the PDX and PDXO biobank, which means that drug developers have more options to find their specific models of interest across different cancer types that represent sort of patient diversity. This will be for both early drug discovery as well as late translational research. There's a vast number of models that we've validated in this paper, but the characterization of the organoids and the PDX are available for access via our online database, which is HuBase for PDX. So this is shown on the left-hand side, and the number of models for different indications is listed. And then organoid base, which is shown on the right. And this is for all our organoids, actually, which includes the PDXOs. So these are ones derived from the PDXs, but also the tumor organoids directly from patients, as well as normal tumor organoids. They're also listed on the right. We've also published in current protocols, and this is a comprehensive review of matched organoid and PDX systems that have been developed not just in our labs but also across different labs, and also discuss the sort of utility and application of these matched models and how organoids can inform on in vivo response, and also how the PDX can be used to create new models. And then we can then use the organoids to create further models because we can actually manipulate them much better in vitro.
And given that, when would you more likely want to perform an experiment using organoids as opposed to xenografts?
So there's many different applications of organoids. and there are many advantages over different systems. So in the current protocols paper, we also summarize the advantages and disadvantages of PDX models as well as the organoids for efficacy evaluation, so that the right platform could be selected for the specific research questions based on its merits. And now the obvious one is the advantages of using it for using organoids for high throughput screening and mimicking a clinical trial in a dish, or maybe better informing on in vivo studies. And here in figure 2E and F, optimizing drug combinations in vitro is another one that's a huge advantage of conducting in vitro with the organoid screens. And you can identify models quickly and which ones will benefit most likely from a combination strategy, but also which models, which dose combinations would be most beneficial. So our team tested the impact of 2 inhibitors with standard care gemcitabine and p53 mutated pancreatic models using a matrix of different drug combinations across the models. And our bioinformatics team was then able to apply mathematical modelling of synergy to determine which dose combination was more effective. And this is shown using a 3D surface plot at the bottom here. And this is the most effective combinations shown in the peaks, which are highlighted in red, compared to maybe potential antagonistic combinations of certain doses, which are shown in the green. And this sort of data is really informative and can be generated quickly across different patient-relevant models and help inform on downstream combination studies, further downstream in combination studies or follow-on in vivo studies.
And lastly, how has this research benefited the oncology field and drug discovery overall?
I mean, PDX originally made a dramatic impact on oncology research, with thousands of models being generated and created to kind of help address some of the shortcomings of other available models, such as the 2D cell lines. However, there are shortfalls of PDX models, such as they are immunodeficient. The time and resources to run in vivo models can be quite costly, and also the sort of lack of in vivo use and manipulation. So if you want to do genetic manipulation, it's really difficult to do with the PDX models. So having PDX in vitro is actually very attractive, but it's not always feasible. So in this paper, we actually show how you can still leverage PDX models to create organoids and complement the in vivo studies with upstream in vitro investigations to refine the in vivo studies, as well as manipulate the tissue in vitro more successfully, and then to use for further in vitro testing and also in vivo modeling. So, for example, when we do graft versus host, when we do humanized mouse experiments, we see a lot of graft versus host disease in the PDX models, which means that it can complicate the data interpretation for in vivo. But in vitro cold culture systems, which is shown here, you can actually circumvent some of these issues well, to an extent. So what we do here is we look at the impact of immune cells co-cultured with the organoids, and then we can also assess immunotherapeutics. And we can use different readouts like tumor kill or cytokine production, and we can also now apply high content imaging and image analysis. So here we show how CAR T cells can be screened in different patient derived models, and we also show how genetic engineering can also support assay development for screening. And like I said before, in line with the recent FDA Modernization Act, this publication is very timely and demonstrates the sort of predictive potential of in vitro testing and the adoption of patient-derived models, as well as obviously PDX now, early in the drug discovery rather than using them later in translational studies. So hopefully I've been able to highlight some of these benefits for oncology drug discovery.
That concludes our session, and we look forward to having you join us for our next publication Q&A session in the very near future.